back to another super cool, super awesome and absolutely amazing session that we're going to be taking right now, right here. Hi everyone. Hello. Good morning once again on this bright, beautiful, clear morning. Good morning, good morning, good morning. How are you doing? Are you all doing good? So today happens to be a Friday, which we are going to be turning into a Friday with a super cool, fun-filled video that I've got for all of you, right? So make sure that you see our amazing Friday video that we're specially getting for all of you. We will also have Bio Bomb. We will also have Vanictionary coming. We will have Biolingo. Yesterday we did Biothon, jinke prizes will be announced in the evening. So because we are doing so much for you, and I also hope that all of you saw a beautiful video which we made with the brave mother of Martyr Major Akshay Girish. I hope you all saw the video, yes, which was telecast on the 2nd of October, that is Gandhi Jayanti. And because we are doing so much for everybody, especially the Friday series and all of this, I want you to let your family and friends know about it. Yes, because it's not only a biology channel, it's a channel which has everything, you know, in it. It's like a total complete dhamal channel. So please allow your family and friends also to witness what beautiful things we do. So please make sure you tell this to your family and friends as well, and ask them to also like, share and subscribe to our amazing channel. Yes? So happy Friday will be our mantra. Yes, everybody. How are you? So should we start today's amazing class with a Menti quiz? Good morning. Should we start today's class with a Menti quiz? Right, tomorrow also we're going to meet sharp at 10 o'clock. Okay? So good morning. Let's start with a Menti. And then we'll come back to biotechnology. For Menti, you need to go to www.menti.com and use the code 82935. Come on. That's the code. Joined everybody? The code happens to be 82935. So that's the code. Come on, joined. Happy Friday. Is it, Abhay? How nice. 82935. Come on, that's the code, everybody. So log on to menti.com, use the code 82935. Don't be late for the class. Biotechnology, very important. So let's start in 5, 4, 3, 2, yes, 1, and we start the quiz. Come on. Any DNA molecule that has the ability to replicate in an appropriate host cell to which the desired genes are integrated for cloning is called as. Come on, good question. Come on. Okay, 15 more seconds remaining. 13 seconds. Yes, 12. 9. Close to 5 seconds. We have a 3, 2, 1, and time is up. Amazing. So, this is what I'm going to be teaching you in today's class. You've all made mistakes. That is why I tell you to attend all classes on time. And you don't attend the classes because you don't want a seat. And the same mistakes you will do next year. Plasmid, Cosmid, PhageMid, BAC, YAC, HAC, plasmid, cosmid, phagemid, bacterial artificial chromosome, yeast artificial chromosome, human artificial chromosome, all of them are what? Vectors. So plasmid, 104 of you have made mistakes. Can you imagine? 104 of you have made a direct mistake. All of them are vectors. So plasmid is not a wrong answer. It's not a wrong answer. No, no, no. But vector is a better answer. Is that clear? In the exam, you can have, dekho, that is why I always say, come here to learn so that you can do well in your examination. Yes or no? इसीलिए मैंने पहला ऑप्शन प्लास्मिड रखा. Honestly बताओ, कितने बच्चों ने पहले प्लास्मिड देखा और बाकी के 3 ऑप्शन पढ़े ही नहीं? Honestly हैंड रेस करो. प्लास्मिड देखा और बाकी के 3 ऑप्शन पढ़े ही नहीं. You didn't even bother to read. Plasmid. This will happen in exam also. Yes, Yash. This will happen in exam also. And let me tell you, कई बार होगा ऑप्शन C is also correct. लिखा होगा प्लास्मिड, ऑप्शन C में लिखा होगा कॉस्मिड और D होगा both the above या all the above या A and B. Is that clear? D में लिखा होगा. So is that a big lesson today that number 1 you have to read all 4 options? You have to read all 4 options. You have to read all 4 options. Come on everybody, you have to read all 4 options and choose the best option. Obviously it is confusing. It is confusing, but vector is a better answer. So should we do one more quiz? First, let us see the leaderboard. Excellent question there. Yes, excellent. So let's look at the leaderboard in today's class. I think Saloni is going to be topping today's leaderboard. So Saloni is topping and she's also the fastest. And then we have Iron Man. And then we have one more Saloni. And then we have Rupa, Kishan, Sumit, Priya, Gurjar, Alisha, Priya, Shalesha and Johnny. Good, yeah, both the Priyas are doing very well. Saloni is doing very well. Where is Kiro? Where are the all regular students who come on our leaderboard? Excellent there. So should we do one more? Should we do one more? That's nice, Rupali. Should we do one more? Yes, on your screen, next question of biotech here. Take your time to solve it. I'm going to move aside so that your attention can be on this. Yes, excellent. Excellent. Come on. True. Come on. You have got about 20 seconds to solve it. Come on. Think before you ink. Think before you ink. 10 more seconds. 8 more seconds. And 5 more seconds. Yes? Lovely. This is good. Good answering. That means you read all 4 options. See, vector के पास origin of replication होना चाहिए. Origin of replication के ऊपर ही it will be able to replicate. The vector is going to carry the gene which I need. The gene which I need is going to be carried by the vector. It needs to have an origin of replication. It needs to have a selectable marker. All of this I am going to teach you in detail. Selectable marker होना चाहिए because on the basis of selectable marker ही मैं decide करूँगी whether it is a transformant or non-transformant. ये सब कुछ मैं आपको पढ़ाने वाली हूँ. Don't worry. It should have unique restriction sites. It should have very unique sites and it should have all of these. All 3 of them are correct. Is it clear? Yes. So let's look at the leaderboard. Menti leaderboard once. Yes, and then we will start our today's super cool class. So I think Saloni is again going to top. Saloni is going to come on top. Good, 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 good. So Saloni, Iron Man, Rupa, Priya Singh Gurjar, Priya Shalesha, Kishan, Medha, Kiro has come back like a crocodile. Uttam and Anjali. Very good. No, plasmid is a vector, but it cannot be only one vector. There could be many types of vector. Right? Done? One more Menti after studying. So first let's study for some time and then we will have one more Menti. Yes? Aapko here, if you don't get marks, it's okay. Here, your own rank should improve. So here, if you don't get marks, that's okay. You have come here to learn. Your main thing is, on this, what is this? On this, you should get marks. What is this? What is this? I will tell you, Khushi. What is this? What is this? When should you get marks? 3rd May. 3rd May. 3 fingers nahi hai, ye 3rd May hai. Yes, on 3rd May you should get marks. Yes, 3rd May 2020. So if you come here and if you learn, you will all get marks on 3rd of May 2020. Yes? Chaliye. So let's start. Dilip, what happened? Why are you making a sad face? So let's come to the process of biotechnology, principles and processes. Lecture 3 in today's class. Very important lecture. No, Ishan, I have explained plasmid is not more specific. Vector is a better answer. Vector can be anything. They have not asked for a specific one. They have not asked for specific carrier. It can be anything. Okay? Yes? Chaliye. So let's start today's super cool class. Did we do the types of restriction enzyme in yesterday? So what is the function of restriction enzyme? Quick recap. Restriction enzymes, those of you joining new should know restriction enzyme. Yes, Yash, thank you so much for answering that. Restriction enzyme cut the DNA. So anything which can cut the DNA are referred to as restriction enzymes. Did I talk about agar-rose gel electrophoresis in yesterday's class? So I told you DNA is negatively charged. DNA is negatively charged. We cut the DNA with the help of restriction endonuclease. The cut DNA is small. The smaller DNA is lighter. The lighter DNA is, moving faster. The shorter molecules, small molecules will move fast. And they will move on a plate of agar-rose gel. So we separate the DNA along the molecular weight. We separate the DNA on the basis of the molecular weight. The cut DNA is small, negatively charged, moves towards a positively charged anode. What you also should remember is, can you see that the DNA is colored orange? Can you all see? This is very important. NEET 2017, DNA is negatively or positively charged. Can you all see that DNA is orange here? Can you all see that DNA is orange? So, yes everybody, we use a dye to see DNA. The dye which we use to see DNA is called as ethidium bromide. Which dye? Ethidium bromide. And when you expose the ethidium bromide to ultraviolet rays, it's a NEET question. It will appear orange in color. Right? Process is called as elution. This process is called elution. MCQ question. Gel, agar-rose gel, MCQ question. DNA will move towards negatively charged. DNA is negatively charged. MCQ question. Will move towards positively charged anode. MCQ question. Yes, there is somebody called Amrendra Bahubali in our class. कल मेरी क्लास में एक नेमार जूनियर था, फिर एक लियोनल मेसी था. आज मेरी क्लास में एक अमरेंद्र बाहुबली है. I don't. Yes, if the DNA is negatively charged, it will move towards a positively charged anode. Yes. So somebody is telling me कटप्पा भी आएगा. Yes, कल मेरी क्लास में एक नेमार जूनियर था, बड़ा मजा आया. फिर नेमार जूनियर को देखकर लियोनल मेसी कहता मैं कैसे पीछे रहूं. Right? So लियोनल मेसी भी आ गया. Then आज कोई अमरेंद्र बाहुबली है. I'm in confusion. और अमरेंद्र बाहुबली को कंफ्यूजन हो रहा है. Yes, अमरेंद्र बाहुबली, बताइए आपका कंफ्यूजन कैसे दूर किया जाए. देव सेना भी है. Yes, everybody. So अच्छा है क्लास के बीच में ऐसे हंस लेना चाहिए. It makes the mood much lighter because we're studying a bigger topic. Yes. रोनाल्डो नोट्स बना रहा था बैठ के कोने में. आयरन मैन भी था आज. Yes. कैप्टन अमेरिका भी आ जाएंगे कल. Yes. बाहुबली, आपको कैसे कंफ्यूजन हो सकता है अमरेंद्र बाहुबली, बताइए. यस, चले स्टार्ट करें. एनी डाउट्स? सो अब शुरू करते हैं क्लोनिंग वेक्टर से क्लास. डन? यस, राइट. तो क्लास के बीच में ऐसे छोटे मोटे जोक बना देने चाहिए बिकॉज इट काइंड ऑफ लाइटन्स द एटमॉस्फेयर ऑफ द क्लास एंड it's easy to understand the class, right everybody? So don't worry, the coming week from Monday to Saturday, the week which is coming, you will see lectures, good lectures taken on timetable, time management, right? You remember the series I'm talking about, you, me and we? You remember you, me and we helpline? I talked about it yesterday. Do you remember that? Come on, just say yes or no, otherwise I'm going to get angry and I'm going to end the class and go. You remember you, me and we? So on that, I'm going to talk about time management. Okay? I'm going to talk about stress. I'm going to talk about 6 months timetable. So all of that I will talk. Every month, every week we will pick up one topic and we will talk about it. Yes, everybody? Time management, solving MCQs, avoiding negative marking, getting up on time. All of this we are going to talk about it in detail. Done? Is that clear? Yes, everybody. So there is going to be one class on time management, one class on how to sleep, what to eat. All of this we are going to have a class. Is that clear? But for that, you have to give me a Diwali gift. Can I ask for a small Diwali gift? For you, me and we. Can I ask for a Diwali gift? Yes, course मैं खत्म करवा दूंगी आपका Feb तक. Done? Can I ask for a small Diwali gift? Making notes, how to make notes and all of that. Can I ask for a small Diwali gift? Can we try and cross 1 lakh by Diwali? Yes? Yeah, because we are working so hard from morning 7 o'clock to evening 12 o'clock. So can we please become this big that we have a silver colored cute gift on Diwali? How to revise an entire chapter in 1 hour. अच्छा, एक चीज है, एक small चीज which I want to request you, one small cute thing I want to request you. In the comment section, can you please tell me most important topics on which you want videos for you, me and we? आपको क्या क्या प्रॉब्लम है, सारी प्रॉब्लम बता दो. डन? किसी को रिलेशनशिप में एडवाइस चाहिए, किसी को टाइम टेबल चाहिए, किसी को टाइम मैनेजमेंट चाहिए, किसी को रिवीजन चाहिए, किसी को नोट्स बनाने का प्रॉब्लम है. कैन यू डू दैट? किसी को हेल्थ इशू है, किसी को पेरेंट्स के साथ प्रॉब्लम है, किसी को गर्लफ्रेंड बॉयफ्रेंड प्रॉब्लम है, किसी को फाइनेंशियल प्रॉब्लम है, किसी को इमोशनल प्रॉब्लम है. सो कैन यू जस्ट राइट डाउन एवरीथिंग, राइट? किसी को हेल्थ इश्यूज हैं, बॉडी इश्यूज हैं, किसी को कॉन्फिडेंस का प्रॉब्लम होता है, किसी को पीयर ग्रुप का प्रेशर है, किसी को ये नहीं पता चल रहा रिपीट करना है या नहीं, कोई बीएससी में है तो वो नीट की तैयारी करे, क्या करे, राइट? ऑल ऑफ दिस कंसंट्रेशन इशू है. सो देयर इज सो मच. Will you just write it down? I promise I will take every issue. That's my promise to you. किसी को रिवीजन का प्रॉब्लम है और किसी को ये सारा प्रॉब्लम है, वो बेचारा बेचारी, किसी के सर पे ये सारा प्रॉब्लम है. यस यस, टाइम टेबल. तो इन द कमेंट सेक्शन आप लिख दोगे स्लीपिंग प्रॉब्लम. उठो कैसे भाई? स्लीपिंग प्रॉब्लम, हाउ टू गेट अप. सारा दिन नींद आती रहती है. किसी को एडिक्शन है टीवी का, टीवी एडिक्शन है. यस, कोई पेंटिंग बनाना चाहता है पर मम्मी पापा कह रहे डॉक्टर बन जाओ. फिजिक्स बिग प्रॉब्लम. मेडिकल स्टूडेंट्स, कोई सिंगिंग करना चाहता है बट मम्मी पापा कह रहे डॉक्टर बन जाओ. So all of this is there. Will you please write it down in the comment section? I promise I will take up each and every one of them. That's our promise to you of the entire team. Done? Done, but you have to write it down in the comment section. So let's start today's super cool, super fun series. The more you write on the comment section, I will address all the problems. Done? Chaliye. So let's start today's amazing class. We've done this and let's come to cloning vectors. Now, this is my gene of interest. Can you see that? Can you all see that? This is my gene of interest. I need this. Suppose this is my gene of interest. Can you see this? I made it with red color only. ये मेरा gene of interest है. I need this. It is my gene of interest. I need this. Is that clear? Suppose it is a gene of insulin. Suppose I want to make glucagon. Suppose I want to make thyroxine. Are you getting my point? Are you getting my point? Ecology and human physiology will be done, Tabassum Hussain. Don't worry, just pay attention in the class. When I'm teaching you something, pay attention in the class. I talk to you, that time you tell me everything. Yes? Is it clear? So pay attention in the class. So gene, तो ये human beings की gene है, human की gene है, human. अब human की gene is very big, so उसमें से insulin का gene is small, so I take out the gene. I just cut and take out the gene. Pin drop silence. Can you see कि insulin की gene के आगे और पीछे? Can you see insulin की gene के आगे पीछे? I have cut it. Can you see I have cut it? I have cut it. Can you see I have cut it? And I take it out. I've taken it out. Kishan Pandey, maintain silence. Right, everybody? I take it out. And now, can you see this is a plasmid? Can you see the green color? Can you see the green color? Can you all see the green color? Abhay, Shaharan, you are the class monitor for today. Yasin Mohammed, you are the class monitor for today. And Dheeraj Kalp, you are the class monitor for today. So Dheeraj Kalp, Yasin Mohammed and Abhay Shaharan. These three people are class monitors, control the class. So I take the gene and I insert it here. So your plasmid is very, very small. If it is small, it can replicate faster. It can replicate faster. So इसके पास एक ORI होगा, उसकी वजह से ये जल्दी जल्दी replicate करेगा. And can you see I will join them? So this is my gene of interest. This is the plasmid. मैंने इनको join कर लिया. When I join this, I get a recombinant vector. It is recombinant. Can you see the vector? Yes? It will recombine. So vector is anything which can carry the foreign DNA. Anything which can carry the foreign DNA is called a vector. Plasmids are generally circular DNA. आपने देखा था वहां they are circular. Can you see this? अब ये देखो अपना कैसे, ये देखो बैक्टीरियोफेज कैसे अपना DNA उसके अंदर इंसर्ट कर रहा है. देखो ये एक वायरस है और ये वायरस कैसे अपना डीएनए इंसर्ट कर रहा है बैक्टीरिया के अंदर. देखो सी, ये देखो देखो वायरस कैसे अपना डीएनए इंसर्ट कर रहा है. सो प्लास्मिड्स, इट्स अ बैक्टीरियोफेज. Bacterio means bacteria, phage means to eat. It is a virus which eats bacteria. It is called a bacteriophage. What is copy number? Copy number एक बड़ा important concept है. ये NEET के exams में बहुत ज़्यादा बार आता है. It's a very very important concept. Copy number मतलब जो भी आपने DNA लिया है या जो भी आपका gene of interest है जो आपको चाहिए, your gene of interest, how many times is it multiplying? Is it multiplying once? Is it multiplying twice? Is it multiplying many times? How many times is it multiplying is the copy number. Is it clear? So कई लोग सिर्फ एक या दो कॉपी बनाते हैं. अब you are doing biotechnology, आपने इतनी बड़ी, you have a big factory. Now when you have a big factory, आपको एक या दो कॉपी चाहिए या आपको millions of copies चाहिए? Tell me. Do you need one or two copies or do you need many copies? You need many copies, large quantities. large quantities, right? And they make many. देखो बेटा कॉपी नंबर क्या होगा? Have you made this? आपने ये बना लिया? Is this your recombinant? Yeah, that's a PCR type. Is this your recombinant? क्या ये आपका recombinant है? Why is it recombinant? Because इसके अंदर वेक्टर है प्लस जीन ऑफ इंटरेस्ट है, जो आपको चाहिए जीन ऑफ इंटरेस्ट, राइट? वेक्ट सो आपको इस रिकॉम्बिनेंट की कितनी कॉपीज चाहिए? एज मेनी एज पॉसिबल. आपको बहुत ज्यादा कॉपीज चाहिए, एज मेनी एज पॉसिबल, दैट मेनी कॉपीज यू वांट. सो इसका कॉपी नंबर बहुत ज्यादा होना चाहिए. इज इट क्लियर? Right, that's about copy number. The copy number should be very, very high. Bacteriophages are also vectors. What are the features of the cloning vector? Very important from this slide. One question can 100% come in the exam. Is it clear? One question can 100% come in the exams. Very, 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 very important. So, सबसे पहले आपको क्या चाहिए? One. Right? One. You need a. सबसे पहले तो देखो आपको origin of replication चाहिए. अगर आपने इसके अंदर फॉरेन जीन डालिया, फॉरेन जीन डाल देते हैं हम इसके अंदर. देखो तो मान लो ये फॉरेन जीन है हमारी, ये फॉरेन जीन है. सो वी नीड रेप्लिकेशन, वी नीड दिस प्लास्मिड टू रेप्लिकेट. ये डिवाइड तो करे, हमें ये चाहिए. सबसे पहले क्या चाहिए? प्लास्मिड रेप्लिकेट करे, ORI. और ORI के साथ ही क्या चाहिए? वेरी वेरी हाई कॉपी नंबर. A very very high copy number. हमें चाहिए कि वो बहुत ज्यादा बार replicate करे. It should replicate many times. Are you getting my point? Are you getting my point? So it should replicate many times. We require a very high copy number. It should divide. Now imagine करो आपके पास दो प्लास्मिड है. I'll make a beautiful diagram. Imagine करो आपके पास दो प्लास्मिड है. एक ये, let's call it सोनम प्लास्मिड, एक और let us call it अमीनूर प्लास्मिड. So we have two plasmids. One plasmid is सोनम प्लास्मिड and one plasmid is अमीनूर प्लास्मिड. तो सोनम प्लास्मिड ऐसा ही है, पर अमीनूर प्लास्मिड ने एक फॉरेन जीन ले लिया है. So who is a recombinant? Who is a recombinant? Who is a recombinant? कौन है recombinant? ये रिकॉम्बिनेंट नहीं है और ये रिकॉम्बिनेंट है. दिस इज अ रिकॉम्बिनेंट प्लास्मिड. अमीनूर प्लास्मिड इज अ रिकॉम्बिनेंट प्लास्मिड. सो आई डोंट वांट सोनम, आई डोंट वांट सोनम, बट आई वांट अमीनूर. यस और नो? अमीनूर भाई है अपना. यस, अमीनूर इज री. सो बेटा सिलेक्टेबल मार्कर बोलेगा सोनम को हटाओ, अमीनूर को लाओ. याद रखना, यही एग्जाम में याद रखना, कितना इजी हो जाएगा. सोनम को हटाओ, अमीनूर को लाओ. क्या बोलेगा? सोनम को हटाओ, अमीनूर को लाओ. इज दैट क्लियर? क्योंकि अमीनूर के पास फॉरेन जीन है. अमीनूर फॉरेन जाके आया है, अमीनूर ऑस्ट्रेलिया घूम के आया है. अमीनूर के पास फॉरेन जीन है. अमीनूर गया था ऑस्ट्रेलिया घूमने, घूम के वापस आ गया. मेंटी को इस 5 मिनट में आने वाला है. इज दैट क्लियर? Is that clear everybody? So selectable marker will help you to choose between, selectable marker will help you to choose between ट्रांसफॉर्मेंट एंड नॉन ट्रांसफॉर्मेंट. तो अमीनूर क्या है? ट्रांसफॉर्म हो चुका है, ट्रांसफॉर्मर बन गया है, ये वो ट्रांसफॉर्मेंट है. इज दैट क्लियर? सोनम रेज योर हैंड्स. अमीनूर रेज. अमीनूर इज देयर ओनली. यस. एंड सोनम क्या है? नॉन ट्रांसफॉर्मेंट. इज दैट क्लियर एवरीबडी? Yes everybody. So transformant is anybody, let's make it more serious now. Transformant is anybody who is having a foreign gene. So the selectable marker will help you to choose between a foreign gene. So it will accept the foreign gene. That's the work. Is that clear? Yes, it will accept the foreign gene. Is that clear everybody? Number 3, vectors for cloning genes in animals and plants. तो हमें ऐसा वेक्टर चाहिए जो प्लांट्स में भी हो, एंड सोनम यही है, और एनिमल्स में भी हो. एंड क्लोनिंग साइट, ऐसी जगह चाहिए जहां पे हम फॉरेन जीन वगैरह डाल सके. मुझे ऐसी जगह चाहिए जहां पे मैं फॉरेन जीन डाल सकूं. आई नीड अ क्लोनिंग साइट. इज दैट क्लियर? Right everybody? So that's about cloning vectors. So we started with this, then we came to the properties of cloning vectors, then we came कैसे insert कर रहा है, and finally we come to cloning में. तो एक एक करके हम सारी properties देखते हैं. Done? एक एक करके हम सारी properties देखते हैं. TI plasmid होता है, तो पहले एक या दो Menti quiz कर लें, और should we do 1 or 2 Menti quiz and then come and see the properties? Should we do 1 or 2 Menti quiz? Yes, everybody. So come back to Menti. So this is the old leaderboard. Remember the old leaderboard, and let me see who's going to top the leaderboard this time. This is the old leaderboard. So let's move ahead. Next quiz on your screens. Yes, 1 second. चलो, come on. The first engineered plasmid vector is, which is the first plasmid vector? Come on, you still have 25 seconds, guys. Come on, is it PBr322, PUC, PSC 101? Come on, come on. 10 seconds. PUC 19. Tell me also in comment section, tell me the full form of PBr322. Tell me the full form of PBr322. Time is up. Is it right? Is it right? Yes? It's the first one to be named, to be found out. Is that clear? Yes? The first one to be named, to be found out, to be given for everybody to be used is plasmid, and plasmid of Bolivar and Rodriguez. But the first one to be genetically engineered, to be modified, this was the first one to be found. Look at the question properly. Right? The first one to be found, the first one to be named is plasmid of Bolivar and Rodriguez, as you are saying, 322, because it was a 3/22 attempt. But the first one to be engineered. Is that clear? So should I start teaching you this? अब पता चल गया क्लास कितनी important है? अब पता चल गया क्लास कितनी important है? Yes, और कल की क्लास में आई एम गोइंग टू स्टार्ट लाइन बाय लाइन NCERT. कल की क्लास में आई एम गोइंग टू स्टार्ट लाइन बाय लाइन NCERT. कल की क्लास में बड़े बड़े बैनर देखोगे NCERT, और मैं आपको ये दिखाऊंगी. Yes, everybody, चलो. So let's move on back. एक बार लीडर बोर्ड देख लेते हैं. चलो पहले लीडर बोर्ड देख लेते हैं, फिर मैं आपको ये दिखाती हूँ कि आज की क्लास में कहाँ पे मैं पढ़ाने वाली हूँ. डन? लीडर बोर्ड देख लेते हैं एक बार. सी एक क्वेश्चन से कितना फर्क पड़ता है. सो वी हैव वर्षा, सलोनी, आयरन मैन, रूपा, प्रिया सिंह गुर्जर, प्रिया शालेशा, किशन, मेधा, कीरो एंड उत्तम जना. गुड, वेरी गुड. तो मैं अभी आपको थोड़ा सा पहले पढ़ा दूं. Yes, everybody, और फिर I will come back to this. Is that clear? Yes, right, दिलीप. मैं एक बार पहले थोड़ा सा पढ़ा दूं and एक बार दोबारा, then I'll come back to it. Yes, पहले देखो अभी क्लास में आगे क्या क्या पढ़ना है. So we have to study in detail about origin of replication. We have to study about selectable markers, कैसे हेल्प करते हैं, cloning sites. राइट, सिलेक्शन कैसे होगा, ट्रांसफॉर्मेंस नॉन ट्रांसफॉर्मेंस कैसे बनेंगे, इंसर्शन इनएक्टिवेशन क्या होता है, ये सारा आपको पढ़ाना है। फिर प्लास्मिड्स पे आऊंगी, कॉम्पिटेंट होस्ट क्या होता है, माइक्रो इंजेक्शन वगैरह बहुत ज्यादा इंपॉर्टेंट है। यस, महिमा वही कह रही है, पीएससी 101 दिया है, नहीं, इट्स पीयूसी स्पीशीज, राइट। तो चलो स्टार्ट करते हैं शुरू से। सो क्लोनिंग वेक्टर्स के फीचर्स कर लिए, एक के बाद एक हम क्लोनिंग वेक्टर्स के फीचर्स करते हैं। यस एवरीबडी, राइट एवरीबडी। चलिए, सो सबसे इंपॉर्टेंट प्रॉपर्टी क्या है क्लोनिंग वेक्टर्स की, मुझे बताई जाए। सबसे इंपॉर्टेंट प्रॉपर्टी, उनके पास ओरिजिन ऑफ रेप्लिकेशन होना चाहिए। इज दैट क्लियर? व्हाट इज द मोस्ट इंपॉर्टेंट प्रॉपर्टी? दे शुड हैव अ ओरिजिन ऑफ रेप्लिकेशन। इज इट क्लियर? दे शुड हैव अ ओरिजिन ऑफ रेप्लिकेशन। तभी उनमें मैंने व्हाई हैव आई एडेड द मटेरियल देयर, मैंने सामान ही इसीलिए डाला है, मैंने सामान ताकि मुझे रेप्लिकेशन मिले। और ओआरआई के साथ मुझे एक और चीज क्या चाहिए, आई नीड अ हाई कॉपी नंबर। अगर कॉपी नंबर ही नहीं होगा तो फिर ऑब्वियसली मेरा उसमें कोई ओरिजिन ऑफ रेप्लिकेशन के आगे मैं जीन डालूंगी, जीन ऑफ इंटरेस्ट, तो मुझे हाई कॉपी नंबर चाहिए सो दैट आई एम एबल टू डू इट। इज दैट क्लियर? Is that clear? Then, right, this is the most important part which I want to show you with the help of this photograph. Most important part, most important. Just look at that photograph, just look at that photograph. This is the most important part. I can give it to you in guarantee next 5 minutes, just pay attention. इसमें से 500%, yes, now also it is NCERT book only which I am teaching you, right? So इसमें से 100% एक question आएगा। बेटा देखो अभी 500%, just understand this last 5 minutes of the class, understand this very important concept which I am trying to tell you. Very, very important concept. Please, last 5 minutes, 500% attention here. Now tell me one thing. What is the most important thing for me? Is the most important thing for me the gene of interest? सबसे important तो यही है ना, मेरे पास gene of interest होना चाहिए. This is the most important thing. I should have a gene of interest, मेरे काम की gene. I should have a gene of interest, मेरे काम की gene. I should have it. Yes or no? ये जीन ऑफ इंटरेस्ट को मैं मीडियम पे डालूंगी एंड मीडियम पे डालने के बाद ऑन दिस जीन ऑफ इंटरेस्ट आई विल सी ग्रोथ। ओके, आई एम गोइंग टू सी ग्रोथ। इज दैट क्लियर? अगर जीन ऑफ इंटरेस्ट प्लास्मिड में नहीं गई है, इट्स नॉट गॉन, सो शुड आई पुट दिस प्लास्मिड ऑन ग्रोथ मीडियम? क्या इस प्लास्मिड को भी मैं ग्रोथ मीडियम पे डाल दूंगी? शुड आई पुट दिस प्लास्मिड ऑन ग्रोथ मीडियम? यू ओनली टेल मी. Should I put this plasmid on growth? No, no, because ये तो growth medium के nutrients खा लेगा. I want to make insulin. Insulin बनाने के लिए how many amino acids are required? 51 amino acids are required. How many amino acids are required for insulin? How many amino acids are required for insulin? 51. So अगर insulin की gene ही नहीं है, write this down, it's very important MCQ question of 51 amino acid. अगर इंसुलिन की जीन ही नहीं है, so should I give it 51 amino acid? ले खा खा खा, should I give it 51 amino acids? अगर इंसुलिन की जीन ही नहीं है, अगर इंसुलिन की जीन ही नहीं है, should, yes, I will also make a list of all the books which you have to read. NCERT में कौन से चैप्टर्स आते हैं, बाहर से कौन से चैप्टर आते हैं, U, Me and We में सब कवर किया जाएगा। ध्यान से भी क्लास सुनो, क्लास में ध्यान दो। सो अगर इंसुलिन की जीन ही नहीं है, व्हाई शुड आई गिव इट 51 अमाइनो एसिड? 21 और 30 होता है। यस, इसीलिए मुझे यहां पे सिर्फ उसी को ग्रो करना है जहां जीन ऑफ इंटरेस्ट है। कैन यू सी दिस? नाउ लुक एट दिस, लेट्स डू इट अगेन, राइट एवरीबडी? सो लेट्स डू दिस अगेन. सो सी, कैन यू सी कॉलोनी? यस एवरीबडी, सो कैन यू सी कॉलोनी? आ जाओ देखो, Can you see colony carrying non-recombinant plasmid? Non-recombinant वो है जिसके पास जीन ऑफ इंटरेस्ट नहीं है, जिसके पास जीन ऑफ इंटरेस्ट नहीं है। मैं कह रही थी आ जाओ, उठो, कमेंट करो, इधर ध्यान दो, पे अटेंशन हियर इन द क्लास। इज इट क्लियर? और इसके पास रिकॉम्बिनेंट प्लास्मिड है, तो मुझे इसको निकालना है और ग्रो करना है और इसको मार देना है। तो मुझे कैसे पता चलेगा कौन जीन ऑफ इंटरेस्ट वाला है और कौन जीन ऑफ इंटरेस्ट वाला नहीं है? थैंक यू यासीन. हाउ विल आई कम टू नो किसके पास जीन ऑफ इंटरेस्ट है एंड किसके पास जीन ऑफ इंटरेस्ट नहीं है? सिलेक्टेबल markers. Is that clear? And this is present on plasmid, and the gene of interest is also given on the plasmid. Any doubts? Any doubts? So the selectable marker are usually antibiotic resistance gene, ampicillin resistant gene, right? Tetracycline resistant gene. These are the selectable markers. So I am going to start tomorrow's class with a video on selectable markers. Is that clear? I am going to start tomorrow's class with a video on selectable markers, color coding of See, non-transformants are not showing any color. See this? Can non-transformant no color? And can you see with non-recombinant vector, with recombinant vector, can you see the colors? Yes, everybody. So इनको मैं हटा दूंगी, जिनके पास कलर नहीं है उसको मैं हटा दूंगी और इनको ग्रो करूंगी. So that is why selectable marker helps me to find out मेरा gene of interest, which I am going to start in tomorrow's class. What do you have to do by the end of today's class, my dear kids? Right? Today's class you have to tell me all that you want to know about you, me and we, which is very, 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 very important. Right? You have to see Friday and another episode of Biolingo or Vanictionary and obviously the Bio Bomb. So starting tomorrow, we are going to show you a video on all of this and even 5 videos on this and 5 videos on photosynthesis. So don't miss tomorrow's class. Please share in the comment section what do you want to know. Friday ka kaam karo please. Right? Or kal ki class specifically NCERT live class. Word by word, point by point, NCERT live class. See you tomorrow. Bye.